Alright, so good day everyone. So this is the second video for the topic evolution. So in this video we will going to discuss the possible ways or possible or some first explanation of how life began here on earth. All right, so even though some explanations here are they did not provide some sort of evidence, or the conclusion is do not support the idea of evolution, but still we will going to discuss some efforts of previous philosophers and scientists in order to unwrap the mysteries regarding the evolution of life here on earth. All right, so again, so let's start this second video. All right, so when did life begin? So some scientists speculate that life began around 4.6 or 3.5, or the Earth itself is 4.6 billion years old. So it is around, according to some scientists, it is around 3.5 billion years ago the life began to emerge here on Earth. So as you can see, the first living organism probably is a bacteria. Yan. So as you can see in this picture, yan, these are the probably during the 3.5 billion years ago, yan, these are the first living organism called stromatolites. Alright, so these things are found in Australia, Australian in a part of Australia, in Australian shore rather. Alright, so these are the first photosynthetic bacteria that are found 3.5 billion years ago. Alright, so they let us with their hardened bodies. These are the stromatolites. Alright, so then when did life begin? Right, when did life begin? So when did life begin? So the evidences of when did life begin starts by studying the metamorphic rocks. And so probably that life existed based on this rock dating that we have right here. Life existed for around 3.85 billion years ago. All right. So how do we find out that the certain age of the rocks that we have right here? So these rocks are the ages of these rocks are obtained by radiometric dating. So we call it radiometric dating. So with the use of carbon 12, carbon 14, and carbon 13, carbon 14, so we be able to trace down how old these rocks are. All right. Next one. Rocks before 4 billion years old are rare and hard to find. Why is it so? Because due to some mechanical weathering, due to weathering and pressure and heat of the sun, so continuous pressure, continuous heat of the sun, weathering, and so rocks might be eroded. Rocks which is 4 billion years old might likely to be eroded as those process continues for until this point. So and not only that, not only weathering, we have heavy bombardment of other stuffs coming from outer space that happened around 3.8 to 4.0 billion years ago. So it truly changes the landscape. So rocks older than 3.8 billion years old might or very rare or hard to find. Yeah. So evidence suggests that life as long as 3.85 billion years ago, and definitely at 3.5 billion years ago. So probably these are the two estimated years in which living things speculated to arise. Yeah. So alright, so there are some ways in order to study how did life began by using living fossils. Alright, so these are living organism, but some scientists speculate that they are already extinct. But the thing here is they are used as their DNA can be a good evidence how their ancestor or how life began in the first place. So, for example, we have a living fossil like the tuatara. So, this is a reptile, right? So, this is an example of living fossil. Another one is the coelacanth. Coelacanth, or rather, so wrong spelling. So, alright. So this is a fish. This is a fish believed to live around several million years ago. It should be extinct, but one organism is found in South Africa. So scientists studied and have an idea on how the DNA of these organisms, or the DNA sequence of these organisms, are arranged. Right? So these are the living fossils. So these are living organism that resembles much likely of their ancestors. Alright? So alright, so one example of another living fossil is the bacteria. Alright, so what is inside the bacteria? Can be studied in order to find out how DNA change over time. Alright, so we have different kinds of bacteria here. We have the bacteria, we have the archaea, and eukaryotic bacteria. Alright, so we have it right there. So technically, these are the group of your bacteria, right? Okay. So when did life begin? So probably on the land, since there is no ozone, no oxygen, so the ultraviolet light destroy all molecular bonds that we know. And what happened there is in some shallow ponds, ingredients of life started to form, like proteins, amino acids, nucleic acids, carbohydrates, something like that. So those shallow ponds, with the action of the ultraviolet rays, which is energy source, something like that, forms certain concentrations of organic chemicals. Right? So some of these organic chemicals can be found on deep sea vents. So what triggers the formation of this organic chemical concentration? The temperature on these deep sea vents. Alright, so basically this is how organic chemical originates. Alright, according to some studies. Alright, next one. So the simplest organism today are those dated 3.5 billion years ago, are remarkable and advanced. Alright, so during those time 3.5 billion years ago, the advanced organism there is the bacteria. So even though they are simple, yet they are already advanced in terms of that timeline. Alright, so now the assumption here is, with the presence of certain conditions, if the conditions are perfect, so life will extensively arise. Alright, so there you go. So this is only an assumption. This only is an assumption. Alright, so here we go. What we have right here, these are the attempts to explain how did life originate and how did life evolve in the first place here on Earth. Alright, so in this, we will discuss this one by one, and we will try to find out what is the loophole, or what are the loopholes of these theories that we have right here. Alright, so first up, we will have the spontaneous generation theory. Alright, so alright, let's click that one. Cannot open specified file, so we'll just open it right here. So alright, so I will just have a flash throwback. Yan, flash throwback of this discussion about the spontaneous generation. So technically, Aristotle proposed the theory of spontaneous generation, also known as the abiogenesis. Alright, so in his discussion, the idea is that the living things arise from non-living matter. So basically, the living things on Earth arise from non-living matter, like rocks, like dust, or sunlight, something like that. Alright, so this idea lasted for around 2,000 years. So why do you think that this idea lasted for around 2,000 years? Why did this idea start? Why did this idea did not disappear during that course? So what is missing here? So what is discovered after 2,000 years? What makes this spontaneous generation alive? Or just like a so, how do we call it in terms of science? So not evident, right? So let's start. So this is Aristotle, and alright, so for centuries, people based their beliefs in the interpretation of what they saw. So there's nothing wrong with observation. So what is wrong there is how do you interpret what you saw. Alright, so going in the world around them without testing their idea. So they just observe what they see, and without testing their ideas, they create their interpretation based on what they say, what they saw. Yeah, so they didn't use, technically they didn't use scientific method. Although the first part of the scientific method is observation, but still you need to test it using an experiment. Alright, to answer your question. Right, and their conclusion were based with untested observation. So you have now an idea of how this spontaneous generation becomes prevalently dominant during that time, 2,000 years ago. Alright, so example of spontaneous generation according to them. So observation, in every year in the spring, the Nile River flooded areas of Egypt along the river, leaving behind nutrient-rich mud that enabled the people to grow that year's crops of food. However, along with the muddy soil, large number of frogs appeared that weren't around in drier times. So, meaning to say, this is the observation. So, you can write down on the comment section what is your conclusion. So, remember, during those times, people did not test their observation. So, just write down in the comment section what is your conclusion about this example number 1 observation that we have right here. Alright, so technically the conclusion is that the muddy soil gave rise to frog. To how? Well, I don't know. That's their observation. Alright, so they observed that frogs are moving in the mud after flooding. Something like that. Alright, so that's their conclusion. Example number 2. In many parts of Europe, medieval farmers stored grains in barns that with thatched roof. Alright, as a roof, thatched roof is in Tagalog sira-sira. Alright, so as the roof aged, it was not uncommon to start leaking. This could lead to spoiled or moldy grain. And of course, there were a lot of mice around it. So again, you can pause this video and then write your comment in the comment section. What is your conclusion regarding this observation? Alright, so what do you think is the conclusion here? So again, people did not test their observation, then they come up to the conclusion that the moldy grain give rise to mice. Alright, so that's how they concluded it without any testing at all. Alright, next one. Example number 3. In the cities centuries ago, there were no sewers, no garbage truck, no electricity, no refrigeration. Sewage flowed down the street and chamber pots and leftover food. So, were thrown into the streets each morning. So, many cities had major rat problems and a disease called bubonic plague. So, this is the observation. So, what will be your conclusion? So, write it down in the comment section. Pause this one and then write it down in the comment section. So, what can be their conclusion? So, during that time, their conclusion was the sewage and garbage becomes a rat. Alright, so the many if you have if you have a lot of garbage, then you have a lot of rats. That's how it's concluded. So again, they concluded this one without testing. So see, these are some examples of spontaneous generation or abiogenesis. Alright, so example number 4. Since there was no refrigerators, the mandatory daily trip of butcher in the shop, especially in the summer, meant battling the flies around the carcasses. Alright, so typically carcasses were hung by their heels, and customers select which chunk the butcher would card off for them. So technically, there were no refrigerators during that time, so the carcasses are hung something like, if I will be able to draw. So this is the feet, and this is the feet. The carcasses or the cow, for example, the cow is hung upside down. Alright, so it is hung upside down in a stick or something like that. So that is basically how the carcasses are arranged. So flies are around the carcasses. So what can be your conclusion here? So again, post this video, write your comment in the comment section. Alright, so what do we have right here? Obviously, their conclusion. So you have a right conclusion. The rotting meat give rise to flies. To how? I don't know. Alright, so probably because those people during their time did not tested this conclusion yet. Alright, they just base it to what they see. So they have an idea that the rotting meat give rise to flies. A lot of flies. Alright, so that's example number 4. Alright, so according to some extent, so these are the recipes for abiogenesis. So kill a young bull, bury it so that the horns protrude from the ground. Then after a month, the beast will fly out of the corpse. So meaning to say that the bull give rise to beasts. So to how, I don't know. Alright, so I cannot answer you. So that is during that time. So people 2,000 years ago can explain this one. Alright, so next one, recipe for mice. So place a dirty shirt with some rag and open pot or barrel containing a few grains of wheat or some wheat bran, and in 21 days mice will appear. There will be adult males and females present, and they will be capable of mating and reproducing more and more mice. Alright, so that's how you create mice, using dirty shirt. Alright, so imagine if you have many dirty shirt, if you have a lot of dirty shirt, you will give rise to a lot of mice after 21 days. Alright, so next one. Now, there are some scientists during that time disprove this spontaneous generation. They tried to disprove this. So, to how? Alright, so the first step is Francesco Redi. So, Francesco Redi is an Italian physician. So, did an experiment with flies with wide-mounted jars containing meat. So, here it goes. So, one meat is put into a jar and closed it. And one meat is put into a jar and then let it open. Alright, so if you have concluded already what will happen here, so you have a right conclusion. So the flies or the maggots will rise up on the meat, and then the flies will be there. So the meat literally give rise to flies, but Redi experiment Redi is not convinced with that experiment. So he tried another experiment, and this time, yan. So, and this time his hypothesis, he still stick on his hypothesis that the rotten meat does not turn into flies. So, it is awkward to think that the meat does turn into flies. Alright, so he observed flies to see that which one maggots would develop. So, in his finding, he found out that if flies was closed with a lid, so adult flies could not get in. So, no maggots develop. In a flask without a lid, maggots were seen in the meat because adult flies had laid eggs and more adult flies soon appeared. So, technically, pag walang, if the flask is closed, there's no flies. If the flask is open, there are flies. Alright, so again, he revised the experiment to verify that particular conclusion, and this time he let the flask open, but he sealed it with a gauze or a cloth. Alright, so, but still open. So, the thing there is, it's, on the first flash, we have the same result as the first one. So, the closed one, obviously, didn't have flies. And the other one with cloth, so the flies attracted to the smell of the rotten meat. But the thing here is the maggot is on the cloth. So, you can pause this video and then you can have your conclusion. Alright, so with that, Reddy's experiment, so it disproved the idea of spontaneous generation for larger organism. But people are not satisfied, or they are not satisfied with this experiment. How about the microscopic organism? It is true for large organism, but how about the microscopic organism? How will they arise? So, yeah, so these are one of the loopholes in Reddy's experiment. Alright, so Francisco Reddy, there he is. Alright, so did Reddy use scientific method? What do you think? Alright, so technically, Reddy used scientific method. Alright, so once you did an experiment to answer your question, you already did scientific method. You don't give a conclusion out of observation. That's a no-no. You need to test that one further in order for your argument to be valid. Alright, so in his case, he did not reject his hypothesis. Instead, he accepted it, and there were some questions lingering, even though people are not satisfied with his experiment. But still, yan, you can revise this experiment and work it on your own, and then you prove it into become a theory. Alright, so that's Reddy use scientific method. Alright, so there you go. Alright, so this is step 1. Alright, so there you go. Okay. Step 2, the hypothesis. Alright, so only flies can make more flies. Alright, technically. Okay, so next, the testing. Alright, so you need a control group. Alright, so you need a control group and you need an experimental group. So your research teacher knows or taught you that one already, so you should know that one. So we have variables here, the independent and the dependent variable. Alright, so next. Yeah, so the independent variable is the sealed jars, and then the dependent variable is the number of flies that will be produced. Alright, so again, so those are the variables. Alright, 4, the data. Alright, so the data confirms that the control group, since it is open widely, so the flies entered and the the eggs are laid, then maggots emerge. Then in the gauze covered, fly on the gauze but not in the jar. Then sealed jars, no maggots or flies on the meat. Alright, so next one. Only flies, okay, so with that particular experiment, only flies can make more flies. In the uncovered jar, flies enters and laid eggs. So obviously that will happen. And in sealed jars, no flies or maggots nor eggs could enter. Thus, none of the flies are seen on those jars. So technically, this disproved the spontaneous generation for larger organisms during that time. So, but they are not satisfied. How about the smaller organisms? Alright, so how to disprove spontaneous generation using microbes. Alright, so Anton van Leeuwenhoek created the simple microscope, and he examined that water. Alright, so he examined the water contains tiny animals like protozoa, fungi, algae, and some sort. Yeah, he called it as animalcules. Animals plus molecules, something like that. Alright, so by the end of 19th century, these organisms were called microbes. So regardless of which, tiny animals, fungi, etc., as long that they live in water, these are called microbes. Alright, so because they are small, rather, yeah. So they are small to see. Right, so this is Anton van Leeuwenhoek and his prototype. Alright, microscope. Alright, so Anton's Leeuwenhoek microscope. So Imagine how hard to use that kind of microscope in order to see the specimen that you are observing. Alright. Alright, so John Needham, he showed that microorganisms flourished in various soup that had been exposed to air. Claimed that there was life force present in the molecules of all inorganic matter, including air and oxygen, that could cause the spontaneous generation to occur. So, meaning to say, during that time that the air itself contains all the necessary ingredient for the microorganism to flourish. Alright, so there you go. That's John Needham. That's his hypothesis during that time. Alright, during his result, during his experiment, people didn't realize that bacteria were already present during Needham. So please take note. Please take note that the bacteria is already there. Alright, it's already there. But people don't have any idea how that happened. Or people don't have any idea that that is possible. Alright, so Needham didn't boil long enough to kill the microbe. So during his experiment, this is what he did. So he did not boil long enough to kill the microbes. Alright, so this is his experiment. Alright, so this is the chicken broth with microbial growth. So he boiled it. So according to the experiment, he didn't boil enough. He just warmed it. He just make it warm, and after several days, alright, so the broth becomes cloudy. The broth becomes cloudy, and since it becomes cloudy, we have now an idea that it contains microbes, since it is cloudy already. So what is missing here? Alright, so you can pause this video and then write your comment in write your answer in the comment section. So what is wrong here? What is wrong here is Needham didn't boil too long, or didn't boil the broth too long. Alright, you just make it warm. Alright, so that's Needham experiment. Lazarus Spallanzani did repeat this experiment. He boiled the soups from an hour, almost an hour, and then melting the slender necks closed. Alright, so he melt the neck of the jars. Alright, he melted this one to become a slender neck, something like this. Alright, so he made this plus something like that. Alright, so the soups remain clear. Later, he broke the seals. The soups become cloudy again. Alright, so did you get the... If you understand how he did the experiment, so it is something like this. Alright, so this is the control. This is the control. So he did open the flask widely. So obviously, the bacteria will enter inside the flask. Next one. So the broth were heated. Alright, so the broth were heated and the flask was sealed. There was no growth, but after opening the flask for several hours or several minutes, there was a growth on it. Alright, so what did he conclude? So what do you think is his conclusion? So write your answers in the comment section before you continue this video. So his conclusion was, alright, so his conclusion was that the air contains life force that allows the microorganism to survive on the broth. Alright, so still the spontaneous generation is still there during that time. So meaning to say, Francisco Redi's experiment is not yet not yet a good source of evidence that spontaneous generation is not true. Alright, so during that time. Okay. Alright, so the theory finally changes during the time. Okay, so who's the scientist behind this game-changing event? So how do microbes arise? Alright, so there's a debate in Paris, so 1860. So that debate was won by Louis Pasteur. Alright, so Louis Pasteur. And how did he won that debate? He won that by doing an experiment disproving the spontaneous generation in microorganisms. Alright, so how did he perform the experiment? So This is Louis Pasteur. Alright, so he is the father of. Alright, so the milk that we drink today, it's because of him. So he's the father of pasteurization, or he's not the father, rather. He's the one who proposed to do what they call pasteurization. So what is this pasteurization all about? How is this process being remarkable? Alright, so Pasteur's problem, the microbes come from cells of organisms on dust particles in the air, not in the air itself. Alright, so this is his hypothesis that the microbes are not or what do we mean by that? So the air did not rise or give rise to microbes. Instead, the microbes are in the air. Alright, so in the air already, but not the air itself. Alright, so that the molecules of air contains microbes already. So he put broth on several S-shaped flasks and he boiled it and placed to several locations. So this is the S-shaped flask. So as you can see, it looks like an Erlenmeyer flask. Alright, so it looks like an Erlenmeyer flask or volumetric, or this is a volumetric flask to be exact, or Florence flask. So he melted the neck of that flask and make it into an S shape. So what do you think is the advantage of putting making an S-shaped flask over here? Alright, so the S-shaped flask filled with broth, and the S-shaped flasks were intended to trap any dust particles that contains bacteria. Alright, so that's technically its purpose. Alright, so in the Pasteur experiment, step number 2, he boiled the flask, and technically, you are correct. The microbes are killed. Alright, in the process. So he boiled it around several hours or more than 1 hour. Alright. So after that, he noticed that the flask was put in the various location after boiling, and he noticed that the dust particles are trapped in the S-shaped region of the flask. Alright, so technically, the dust is in there, but the broth did not become cloudy. So how's that? So it means that there is something on the dust that makes the broth becomes cloudy. Alright, so in the dust that we have right here, so microbes, according to him, microbes were not found in the broth. So technically, there's no microbes on the broth. Alright, so notice the dust that collected. So he noticed this one, and this does not affect the broth itself. So here you go. Alright, so now he did repeat the experiment, but this time he broke the stem of the flask, and then he allowed it to be opened, widely opened on air, and then he wait for several hours, and then there's our microbial growth after that waiting period. So what does it mean? It means that, alright, so it proves only that microbes come from other microbes, that life comes from life. So if you notice the cell theory, there are 3 assumptions there. So one of those assumptions is cells can only give rise to a living cell can only give rise to a living cell, and that notion comes from Louis Pasteur. So hence he disproved the biogenesis, or a biogenesis assumption, so that all living things come from non-living things. Now, because of this experiment, this is a game changer. So after 2,000 years, the abiogenesis assumption was completely turned down, and that the idea of biogenesis, that all living cells is capable of giving rise to a living cell. Alright, so this is the biogenesis and abiogenesis theory. Alright, so in review, alright, so Francisco Redi's experiment, evidences. Alright, so these are the questions. So you can answer it on your own. So where does the maggot came from? Alright, what was the purpose of the seal jars? Alright, spontaneous or biogenesis? So, alright, so the answer here is biogenesis. Alright, next one. John Needham experiment. Nutrient broth heated, then placed then placed in a sealed flask. Alright, so the result is microbial growth. Alright, so where did microbes come from? Alright, so is it spontaneous or biogenesis? Alright, so the answer is biogenesis, or rather spontaneous generation. That's according to him. So sorry for that. So a spontaneous generation. And next one. Alright, Spallanzani experiment. He boiled down the nutrient solution in a flask. Okay, so the nutrient broth is in the flask and then heated, then sealed. The result was no microbial growth. Alright, so he sealed the flask. So is it spontaneous or biogenesis? Again, during his time, so Lazaro Spallanzani found out this is a biogenesis. Yeah, so alright, so and finally we have Louis Pasteur. So Louis Pasteur demonstrated that microorganisms are present in the air. Alright, so during his time, the nutrient broth is placed in flask and heated but not sealed. Obviously, microbial growth will happen on it. But the nutrient broth placed in flask and then sealed it with an S-shaped flask. So obviously, there is no microbial growth. Then he concluded that, what do you think? Spontaneous or biogenesis? Alright, so that is biogenesis. Alright, so in this part, so we discuss the spontaneous generation and the difference between the biogenesis. So as you can see, the one that we believe right now is biogenesis, that cells or living cells comes from also a living cells. So what do we have right here? So did biogenesis explain what is the origin of life? So technically, it is somehow yes. All right. So it is somehow yes. He did explain the origin of life. So meaning to say that life came from obviously living things. So if cells that we have right now obviously comes from a living cell. So that's how spontaneous generation theory was turned down during that time, and abiogenesis is the one that we believe right now. Alright, so in this case, so that ends up our first part of discussion about the origin and evolution of life, the spontaneous generation theory. So in the next video, we will tackle the chemical evolution and the symbiont hypothesis, panspermia, and Lamarck's theory of evolution. Alright, so that's all for now. Good day and God bless.